The overall goal of this labeling methodology is to measure synaptic vesicle endocytosis.
This method can help answer key questions in the neuroscience field, such as the dispersion of formed endocytic vesicles. The main advantage of this technique is the resolution during imaging and the visualization of changes by examining the complete structure of a cell. Generally, individuals new to this method will struggle because they fail to remove tiny air bubbles while preparing the epoxy or dry out the sample during the ethanol washes.
To begin this procedure, prepare a poly-D-lysine-coated 6-well plate by applying 1.5 milliliters of 0.01% sterile filtered poly-D-lysine solution to each well for 1 hour at room temperature. Then washing it 3 times with sterilized water. Next, prepare a high-potassium stimulation solution with horseradish peroxidase, and adjust the solution to pH 7.4 with 5 molar sodium hydroxide. After that, stimulate the hippocampal neuron culture by adding 1.5 milliliters of high-potassium stimulation solution to each well at room temperature for 90 seconds. In this step, prepare a 0.1 molar sodium cacodylate buffer at pH 7.4. Fix the cells with 4% glutaraldehyde in 0.1 molar sodium cacodylate buffer for at least 1 hour at room temperature.